Welcome to this video on registration. My name is Mark Jenkinson, and in this talk I'll be talking about image spaces and spatial transformations, which are two of the concepts which are important to understand when you're using registration so that you can choose the best options and get the registration to work the best for your particular problem. There are actually four main concepts that you need to understand, and so we'll be tackling the first two within this talk and then the other two within another talk. So image spaces is the first thing that I'll talk about. And the most familiar one is standard space. So you may have come across Talairach space or MNI space before. That is an average space that we use, which represents an average anatomy, which we use in order to register lots of different individuals to that, so that we can have a common way of describing where things are within the brain. So we use coordinates which map to specific anatomical locations. And that was originally done by Talairach and Turnow on one particular postmortem brain. They did a very detailed dissection and recorded coordinates and talked about how to make that a standardized thing, and that was used in the neuroimaging community at the beginning. Now it's much more common that we use the MNI templates, also called the MNI 152 or the ICBM 152, which is an average of MRI images from 152 young healthy individuals which have been registered together in order to make a template which represents what's common across that population. And you'll see within those images that parts of the image are very sharp. So if you look at the insula, some of these folds here are very sharp. The ventricles, many of the subcortical structures are sharp. But then there are other areas such as the parietal lobes where they're not sharp. And that represents the fact that there's not such a consistent folding pattern across individuals. And when there's not a consistent folding pattern, often the registration can't find a consistent way of registering two different individuals together or an individual to the template, which is how the template is created. And that's because if you've got two folds in one individual and one fold in another one, then there's no way that we can figure out just from the T1 weighted images alone what is the right way to register them. Should you try and unfold the two folds to make it into one? Should you try and add an extra fold into the one that only has one? We don't know what the right answer is. We would need more information to know how to actually map that well, and that's a research project which is going on in the field. But generally, if we're just working with T1 weighted images, we don't know, and that's represented by the fact that in those areas, in these standard space templates, you see that it's a little bit more blurry. So as I said, the MNI 152 is the one that we would generally use. It's close to Talairach, but it's not exactly the same. So if you're looking at very old papers, or occasionally you can see ones now which talk about Talairach coordinates, they are slightly different from MNI coordinates, and just be aware of that. MNI space is also the standard space where many of the atlases exist. So here's just an example of some of the atlases. Many of these are included within the FCell package, which you can find, and most of them are already in the standard space, in this MNI space. That doesn't mean that they've been derived from the same 152 individuals. Generally, they have not. They've been derived from other sources of information, other groups, and may have other demographics. But then they have been registered into and aligned with the standard space templates in the MNI space, and so that that atlas is available with these common coordinate systems so that we can know how to describe it. We also have the template so that we can get other images that we're interested in, that we want to analyze, into that space and then use the atlas information in that way. So both in the creation of the atlases and in the use of the atlases, we are going to need to get our images into the standard space. And so it's a very common thing that we need to do, is we need to move images into standard space, or we need to move the information from the atlas out of standard space into the space of our particular image. And so you've heard me now talking about other spaces, and that's because standard space is only one of the spaces that we have. So if we have a functional imaging experiment, we would have a functional image, a structural image, and then we would want to be getting to standard space eventually. Both the functional image and the structural image are going to have their own space. We're going to talk about them having a space because there might have been a change in the orientation of the head between the acquisitions, or in fact, actually the structural might have been taken in a different session. And so that person might have been out of the scanner and then pulled back in the scanner. because the mapping of the coordinates within those images to the anatomy is different, we describe that as a different space. And we want to be able to move things between the different spaces. So we would start with these 3 different spaces in this kind of experiment. Each of them can have different resolutions. So the standard space, actually, we have templates at 1 millimeter resolution, or 2 millimeters, or 0.5 millimeters, quite common. And so just because the resolution is different doesn't mean that it's in a different space. As long as the coordinate that we would associate with a particular location in the anatomy is consistent, then we would be in the same space. And it's crucial for us to be able to move things between spaces, and that's where registration is used. So we need our registration to be able to link the different spaces together, to align the images from one space to another. And then once we've done that, we can transform information. So imagine that we are interested in the caudate nucleus, and we've got our little mask here. You can see that ROI in red in the standard space. If I've done a registration with our structural image, then I can transform that mask into the structural space, and I can overlay it on the structural image. If I've got a registration to the functional image, then I can also transform it into that space. If I've got 2 registrations performed, then I can actually do everything that I want, because I can easily calculate inverse transforms and move in the other direction. And so in this case, if I created 2 registrations and did 2 different alignments, I would be able to move between any 2 spaces that I want to do. And the FSL tools will often be moving things between spaces behind the scenes for you. And when you look at outputs, you'll notice that some are in certain spaces and some are in other spaces. And you need to recognize that. And if the information that you want isn't in the space that you need it to be in, you need to know how to transform it into one of the different spaces. And so that's why it's important that you understand this concept, so that when you're presented with a set of files and a set of different images, you know how to recognize what things are and why things have different resolutions, they have different fields of view, because that's going to represent that they're in different spaces typically. I've already briefly mentioned the word coordinates before, and I think most people have an idea of what coordinates mean, but actually there are 2 different types of coordinates that we often talk about. So image coordinates, or voxel coordinates, are ones which describe the location within the image in terms of like a row and column. So they're whole numbers. They would start at 0 and count up to n-1. So if my image had 200 voxels on one axis, then it would start at 0 and go up to 199. The origin for this coordinate system is 0, 0, 0, and is in the corner of the image. The axes don't have to have anything to do with the anatomical axes of the brain. It just depends on how that subject was scanned. If you had oblique slices, then none of these axes are going to align with any of the interesting anatomical axes that we're interested in. If you look at the viewers, such as fossil eyes, then they will report these coordinates. And so if you look at fossil eyes, it's actually the second column. It has voxel location written above it, so that you can see. And the location panel is typically in the center of the bottom part of the window. FSL voxel coordinates are the same as NIfTI voxel coordinates. So if you're working with other packages, then, and they're also reporting NIfTI coordinates, then they're going to be completely consistent with what we use in FSL. So sometimes we use these coordinates and report where things are based on these coordinates. But actually a lot of the time we would prefer to use standard space coordinates, particularly if we're reporting things in papers, because that's actually what's common. We use standard space so that we can have coordinates which have meaning attached to where they are in the anatomy of the brain, so that everybody, in this case we have a coordinate minus 0.5, minus 17.5 and 18.5, and you can look that up. You can look at where that is within the MNI, and everybody can do the same thing and know where that is within the brain. So that's the usefulness of standard space coordinates. The coordinates themselves have actual units, so they're in units of millimeters. The origin now is at a specific point, so it's at the anterior commissure of our brain. And that's true for both MNI and for Talairac. And the axes are also aligned with the interesting anatomical axes. That is, left-right is the x-axis, anterior-posterior is the y-axis, and inferior-superior is the z-axis. There are also different standard spaces, so I mentioned MNI and Talairac already. There's also BrainWeb, which was something which you can find, and there are others, but they're far less common. MNI is the most common one, but you don't always know that you have M&I coordinates, because you have to have done a registration in order to establish the correspondence with M&I space before you can even do anything. So when you look at fossilize, the left-hand part of that location panel gives you coordinates in M&I space if it knows about it. So at the very top of that, you can see that it actually says coordinates M&I 152, and that's crucial. If it says that, fine, then the next three values are actually the coordinates in M&I space. If it does not say M&I 152 in this location, then it's probably just scaled up the voxel coordinates. It might say something like aligned and nat, or anatomical, or scanner space. There are various different ways that it can also report some kind of millimeter coordinates. But unless it's actually labeled as M&I, then it's not actually M&I coordinates. So just be aware of that. And it really can only do M&I coordinates if a registration's already been run. So if you've got your scan immediately off the scanner, it will not be M&I coordinates at that point. So that's image spaces. It's a little bit abstract, but it's important to understand when you're looking at the images, you're looking at the outputs, that some of them will be in different spaces, and then often you need to know how to transform them yourself between different spaces. Although the tools are going to do most of that automatically for you, it's not uncommon that you want to do something a little bit different from the default outputs that you're getting. And at that point, it's really important to know about these different spaces and how to manipulate things. The next thing I'll talk about is spatial transformations. So whenever we align images, we have to decide what we're going to allow the registration to do. What kind of things are permissible within the alignment? Is it allowed to change the shape and size of the brain or not? And spatial transformations are how we specify that. They're specified by name and also often by degrees of freedom, or DOF. So the rigid body has 6 degrees of freedom in 3 dimensions, affine has 12, nonlinear has many degrees of freedom typically. And we'll look at these specific examples come out because they're the most common ones. When you look at the GUIs, this specification will be written in various different options that you've got. Often they'll be filled in by default, and if you're using the tool in the default way, then that's fine. But if you're doing anything slightly different, then you need to be very careful that you've selected the right kind of spatial transform here. And so it's an option here because there are lots of things that we do within imaging. There are lots of different ways that you can run imaging experiments, lots of different things that people want to be able to do. And that's why we have these options and possibilities for configuring the tools in that way. And it's why you need to understand when to use different options. So rigid body is fairly straightforward. Our 6 degrees of freedom in 3 dimensions include 3 rotations. So you've got the one here which is about the z-axis. There's also one about the x-axis and about the y-axis. So this is kind of shaking the head, there's also nodding the head, and there's also tilting the head from side to side. And then there are 3 translations. So again, in x, y, and z axes, we can have different translations. And that describes everything that we can do with a physical object without changing its size or shape. And that's actually used normally for within subject registrations, because if you've got a subject in the scanner and they're moving their head about, this is all that they're doing to their brain. They're not changing the size or the shape of their brain, they're just changing where it is in space. So they're changing the orientation, the rotations, and they're changing the position on the translations. Affine transformations take the 6 degrees of freedom that we had from rigid body and add another 6. So in this case, we go up to 12 degrees of freedom. The extra ones that we have are scalings in 3 different axes, so you can see 2 of them here, and skews or shears, which look a little bit like rotations when they're small, but are different because if you look at the top and bottom in this case, it doesn't move up and down, it just moves from side to side. Whereas if I was rotating, there'd be a combination of it moving from side to side and moving up and down. So they're a little bit different, and their skews or shears are the same things, so they're just alternative names, and together we have 12 degrees of freedom in total, and we use those either for correcting for distortions which are linear. So affine is as many degrees of freedom as we can have and still be a linear transform, and this is something which occurs in things like eddy current distortions. They're predominantly linear, but also we use it crucially, and this is the most common case, in initializing nonlinear registration. So we use it in order to get the right orientation and size and rough shape in order to make a starting point for the nonlinear registration. And then the nonlinear can do lots of things. So the nonlinear is local. So if we look at this example, you've got red edges from the image that we're trying to align to, and then we've got the grayscale of the image that we're starting with. And that is being, in general, shrunk down to match the outline of the brain from the red edges. But at the same time, the ventricles are being expanded. And so that's something which you can do when you've got a local transformation which is nonlinear. If it was a linear transformation, it would have to do the same thing everywhere. So if I scaled one part, I'd have to scale every part of the image in exactly the same way. Here, it's nonlinear, it can do different things in different locations. We try and constrain it a bit so that it can't do wild, crazy things. So we don't want it to break up pieces of the anatomy and fragment them and put them in very different places. We want things which stay together. And we also don't want things to disappear. So we normally put some things, so basis functions or regularization or topology preservation or diffeomorphisms are all kinds of constraints. You don't need to know the details about what they are. But you'll see that these are words that come up when you look at nonlinear transformations. They're all just ways of trying to constrain it so that it does biologically meaningful changes, reasonable, plausible changes in terms of how it's changing the shape of the brain, because it's really able to change the shape at quite a fine level. And so we have to use that when we're looking at good quality between subject registrations. Because the shape of different structures, the size of the ventricles, the folding patterns in the cortex are all different. And we need that level of flexibility in order to get good alignments between our different individuals or between an individual and a template. So here's just an example of what these different registrations look like. So on the top row, we start with 5 different individuals. And you can see that they have different sizes and shapes of the brain. The ventricles are quite different sizes. And then on the second row, after linear registration, you can see that they are all oriented in the same way and they all have roughly the overall same size and rough shape. And that's as a consequence of doing our affine registration. And we're doing that to our standards-based template, so our M&I template, which is what you can see on the bottom right. After we've done nonlinear registration, we get what we see in the bottom row. And now, actually, there's very little anatomical difference between all of these different ones. If you look carefully, you still can see that there is some anatomical differences. And as I said, things like number of folds is not going to be fixed if it's different between different individuals. But even things like the ventricles are still slightly different in size. So nonlinear registration is not perfect. Basically, no analysis tools are perfect. But there are some slight differences which remain. But the vast bulk of that change has gone. So if we pick a location within one of these images and we look at the same location in the other images, we are going to be very, very close to the same tissue. And that allows us then to do our group analyses and to look at what's happening at this location within the brain, this sort of anatomical locale. And so that gives you an idea of what the different registrations can do. So when should you use what kind of transformation? Well, we've already talked about rigid body, 6 degrees of freedom. We use that for within subject motion. And so that's typically for doing motion correction within a functional series, or if we're trying to register 2 different images, so it might be a T2 weighted image and a T1 weighted image of the same individual, where they've got the same anatomy and all that they've done is that they've shifted their head about. So if it's the same person within subject motion, rigid body is the best thing that we can do. It's the most appropriate transformation. It provides the appropriate constraints for that. We don't want it to be more flexible. If it is more flexible, then the registration might start changing the shape of some parts simply due to noise or artifacts which are present in the image, which are not due to actual biology. So we like to have the appropriate degrees of freedom and to constrain it in that way. So rigid body is the right thing for within subject motion. For nonlinear, we use that when we've got different anatomies. So we've got different individuals and we want to actually register them together and to actually get the anatomy as best aligned as we can. We use affine in order to initialize the nonlinear. So the first thing that we would do if we're interested in doing 2 different subjects together or a subject with a template is we would do an affine registration first, like we saw in the previous slide, and then we would follow up with a nonlinear registration. There's also a global scaling one, which is 7 degrees of freedom, which I haven't really talked about here. It's basically rigid body plus a global scaling, a scaling which is equal in all of the 3 different axes. And we use that occasionally to correct for drift in longitudinal studies, but it's not very common. But I've just mentioned it here so that you're familiar with that term in case you see it. And remember that we do use the most appropriate one, and it's not the case that more degrees of freedom is better. It is in statistics. We always want more degrees of freedom. It gives us better statistics. For registration, that's not true. More degrees of freedom is not better. We want the appropriate one. So if it's within subject, then 6 degrees of freedom is the best thing that we can do. In terms of the transforms, we store them in different ways. So if we're doing a 6 degrees of freedom or a 12 degrees of freedom linear transform, then we can represent it with a matrix. The matrix looks like you can see on the left-hand side there. It's a 4x4 matrix. The bottom row is always 0, 0, 0, 1. And it's the top 12 numbers which are interesting. And we typically just store that in a little text file. So if you see a text file with this kind of array of numbers, then you know that that is a transformation matrix. It might represent a 6 degrees of freedom, or it might represent a 12. The fact that there are 12 non-zero numbers does not mean that it's 12 degrees of freedom. It could still be 6 degrees of freedom. But either of those are going to be stored in little text files as those matrices. For nonlinear transformations, we can go up to millions of degrees of freedom because actually each voxel can theoretically move independently. We try and constrain it so it's not totally independent, but nonetheless, we actually need to describe what's happening to every voxel. And so we store that as an image instead. And so if you see an image, it'll be a 4D image because actually we need to store 3 components of the vector at each location. So at each location we're going to store how that voxel is moving, which means that we need an X and a Y and a Z displacement. And so we store those 3 components of the vector as a 4D image. So that's 3 lots of 3D images put together. So there'll be one nifty image, it'll be a 4D image, but it will only have 3 components in the 4th dimension. And that is what we describe as a deformation field, or a vector field, or a warp field. We normally use the word warp in FSL, but all of these terms are interchangeable. So if you see images, files like that, then that's what they represent. If you load them into Fossilize and you turn the line option on, then you might see something like you're seeing there. This is a zoomed in part of just a small section of an image. If you don't turn the line option on, it will just look like a sort of weird blurry mess. It will not look like a brain at all. So these do not look like brain images when you load them up. So just be aware, most of the time they'll have the word warp in the file name, which will give you the strongest indication. And then I've talked about the fact that nonlinear can have many, many degrees of freedom. But often we want to control that because we don't want it to be able to do crazy things, and we want it to be able to adapt appropriately to the level of information that we have. I mentioned the fact that our templates are blurry in some areas, so we actually can't drive it that precisely in those areas. And so it's good for us to impose some level of control or smoothness on the warps that we're getting out, so that points that start together stay together. That is, if they're in the same neighborhood, then they'll remain in the same neighborhood, and it won't be able to pull things and distort the local anatomy too much. And there are various different ways that we can look at smoothness. A smoother image will have less degrees of freedom, but it can also be described by having a lower warp resolution. So we often have control points, and the spacing of our control points is one way of controlling the smoothness. And this is actually how we specify it in FSL. So you'll see that there's a control point spacing or a warp resolution. And that's what the red dots here represent. They represent individual points of control. It doesn't mean that that's the error that we would expect, far from it. There's a lot of information about those points still, but it just gives us an idea of what is a local neighborhood and how much we try to keep things together. And also a higher regularization, which is one of the options that you can use within the registration tools, can also create a smoother warp field. To give you an idea of what I mean by a smoother warp field, here's an example where we've got the input image on the left at the top, and we've got our grid that starts off perfectly rectangular. And below that we have the M&I template which we're trying to register to. So we're trying to register the grayscale image above to the M&I template below that. The registration is going to warp that input image, and so we can see that both as in terms of what it does to the grayscale image itself, but also what it does to the grid. And so here on the top row, we see two different options for how it's done the warping. First of all, with a high regularization, that is a fairly smooth thing. And you can see that none of the grid squares have changed that much in size. There is a change in position of things, and you can see that, but it hasn't gone very far away from where it started. Whereas when we lower the regularization, we allow it to be less smooth, then you can see that in one particular place, it's done a lot of changes. So around this area, in this particular sulcus, it's moved things a lot. And the reason for that is that the original sulcus was quite wavy or quite bendy, whereas actually in the M&I it was quite straight and quite vertical. And in the lower regularization option, it's turned it into something which is much straighter than it is when it's smoother. What is the right thing to do? That's hard to know depending on the particular problem that you're trying to solve. And so that's why we have different options for how smooth we want things. But this gives you an idea. If we lowered that regularization even further, it would have even more flexibility to move the anatomy around. And often that is a bad thing because it moves it around in ways which are not particularly biologically plausible. So by default, we recommend 10 millimeters as a control point spacing or warp resolution if you're registering your image to the M&I space. And that's our strong recommendation. We would say stick with that. If you've got two individual subjects, then you could do with less regularization. That is, you could make your warp less smooth. You could give it more freedom to move things around because you won't have these blurry areas within the template where there's less information. You could actually drive it with higher quality information if you've just got two individual scans. And so in that case, we would say, it is an option to try and change that warp resolution. If you make that lower, then you are giving it more flexibility. You're giving it more options to change the anatomy. But if you're just registering to the M&I 152, we strongly recommend just sticking at the 10 millimeters. And that's what I wanted to say. So standard space, is a very common space. M&I 152 is the one that we would normally use. Most of the atlases that you'll come across are already in that space. And therefore we want to move images in between different spaces. We want to be able to get to the standard space for when we're doing group analyses or when we want to take information from an atlas and use it in our particular image. We want to take things from that standard space and take them back into the space of our particular subject image. For all of that, we need registration to be running. There are two different types of coordinates, voxel coordinates and millimeter or standard coordinates, and you can see those in the viewing tools. When it comes to transformations, you need to choose the appropriate spatial transformation for the kind of image problem that you're trying to solve, the kind of alignment. Rigid is the most appropriate one and the best thing to do if you've got within subject. Nonlinear is what you need if you've got between subject and you want to change the anatomy, but you need to do an affine before the nonlinear. That needs to be done first to initialize the nonlinear because it makes it a lot easier for the nonlinear if everything is roughly in the right position to start with. The nonlinear works a lot better when you do it that way around. And regularization is an option that we have which alters the flexibility or the smoothness of the warp fields for the nonlinear. We have a recommendation if you're going to the M&I 152 space, but it's quite common that you might want to do other things in other points in different experiments, different points in your career. And so be aware that that is an option that's available to you in order to adapt the nonlinear registration for your particular purposes.